focus on today in this presentation is some of what we call the neurobiological data collection. And so when looking at a new treatment for Alzheimer's, obviously the most important thing is whether it is effective, whether it can improve people's memory, whether it can improve their quality of life. Now, one of the things I'm really excited about this study is that we are collecting some information, this sort of neurobiological data, that is going to be extremely valuable and really, really helpful in terms of understanding Alzheimer's, but also helping us to determine in future who might benefit the most from TMS. So the way the clinical trial has been designed, we have this longitudinal data collection before and after the TMS treatment. So that's separated by about 13 weeks, so before and after. We're using a comprehensive suite of neuroimaging techniques. So not just relying on one of them, but actually 3 different types, which I'll go through a bit later. In addition to this, one of the really exciting cutting edge factors we've included is this blood test before and after the treatment. So we can use blood-based genotyping and biomarker analysis, which can tell us a lot about someone's symptoms, their Alzheimer's disease pathology, and there'll be some other functions we'll speak about as well. So one of the things I was saying is that this information has a very broad impact and something that is very exciting for multiple purposes. So this neurobiological data, firstly, is something that could potentially be used to predict who would benefit the most or respond best to TMS treatment. Now this is really important, so we know with things like medication, some people may respond, other people may not, and it can be a bit of a roll of the dice. What we have found is that this sort of neurobiological data may be really invaluable for identifying those who are at a much higher likelihood of responding to treatment. Also, in a more broad sense, the information will be a very comprehensive characterization of a large sample of people with Alzheimer's disease. So this kind of information can help us understand the driving factors that lead to symptoms in Alzheimer's disease. So in particular, that sort of communication within the brain that Kate mentioned. And finally, this sort of data could potentially be linked to, as we're saying, the memory problems, to see what patterns of brain activity or what symptoms within blood tests could actually help us potentially predict who may have these symptoms and how severe they may be. So beginning with neuroimaging, so the name of it there, you're probably all familiar with different forms of neuroimaging, or have at least heard about them. So these are ways that we can record brain activity from different individuals, and a number of different techniques are available. We're using what we call a multimodal approach, and what this means is, rather than just relying on one technique, we've incorporated recording from multiple different types of neuroimaging. So the first of these is functional magnetic resonance imaging, or fMRI, which many of you may have heard of. The second is something called electroencephalography, which I'm going to call EEG, because otherwise it's a very big mouthful. And then functional near-infrared spectroscopy, or FNIRS. So what I'm going to do is go through each one of these and just get a bit of a picture about what this can tell us about the brain, and in particular in the case of Alzheimer's. So I find this to be one of the most exciting parts of the study, because by combining these different techniques, we can provide a very comprehensive characterization of brain activity. With each of these techniques having their own strengths and weaknesses, they can actually complement one another. You may ask why research doesn't always just include all of them, and it's because it's quite complicated. There's equipment required, there's technical expertise, and there's also some statistical and computational skills that are required to look at them. And so this is one of the great things with coming to the Bionics Institute, is that we're uniquely suited for doing this kind of approach. They have these electrically shielded and soundproof booths where you can record these different techniques. They also have the EEG and FNIRS equipment. So it really means that the information we gather can be something that is truly unique and really, really valuable. So first off, I'll talk about functional magnetic resonance imaging. So this is what the machine looks like down the bottom left. So essentially, this technique uses strong magnetic fields to look at changes in blood concentration in the brain. And this tells us information about how active certain parts of the brain are. one of the big advantages of this technique is it can provide extremely highly detailed pictures of brain activity. So looking at down to 1 millimeter parts of the brain. So this can be, incredibly detailed, particularly in things like Alzheimer's, where different parts of the brain for different people may be affected. This sort of fine-grained detail could be really valuable. The other important thing is that there are a few decades of research showing that Alzheimer's disease is associated with changes in brain activity when you're using fMRI. So there are these, what we call functional networks, that play a really important role in day-to-day life and in cognitive functioning that are affected in Alzheimer's disease. And so this is a really valuable way to look at those functional networks and how they may be affected. Then looking at EEG, so the brain produces electrical signals, and if you put electrodes on the head, you can actually record the strength of those electrical signals. So that's what EEG does. It records the electrical activity produced by the brain just by placing lots of different electrodes on the head to record that. So in this picture down the bottom, you can see there's a cap on the head and you can just attach those electrodes. Now, the big advantage of EEG is that it is extremely fast. It can be recording brain activity that's occurring up to 10,000 times a second. Now, this is very important because the brain also works very, very quickly. With something like fMRI, it might take a picture once every second or so. Whereas something like 10,000 times a second, you can imagine that you're getting some very, very detailed information. And when you actually process this information, you can get a kind of a breakdown of how brain activity differs between different parts of the brain. And so this is some of the real data we have here, and when it is processed, this is how it can look. Now, what I was saying about this rapid brain activity, so EEG has been around for more than 100 years now, and so there's a lot of research showing that when the brain works, there is this rhythmic brain activity that's really important to support cognitive functioning. So I won't go through all of these, but there are these oscillations occurring around 4 to 7 times a second that are important with attention. Some faster oscillations, so 8 or 12 times a second, or it might be important for ignoring distractions and things like that. And these are really important for healthy cognitive functioning. And so looking at these, we may get some information about how breakdown in this communication could potentially play a role in something like Alzheimer's disease. And again, looking at this, so something like fMRI, which is really detailed but can only record once a second, it can't assess these things that are occurring 50 times a second. It's simply not fast enough to do that. And so this was a paper from a figure from one of Kate's papers, sort of showing how these different regions of the brain may be affected using EEG to look at this activity. And finally, functional near-infrared spectroscopy. So this uses near-infrared light sources and detectors placed on the head to record changes in blood oxygenation. So if anyone's ever been to a hospital and they put a little clip on your finger with a red light, it's essentially doing something similar to that. Shines a light in and it can record how much oxygen is in certain regions. When you actually analyze this, you can start getting information about brain activity in certain parts of the brain. Now, fMRI really fits in the middle of sort of fMRI and EEG. So it can record faster than fMRI. It can do around 10 times a second. And it has more spatial or sort of finely tuned data as compared to EEG. So it can sort of link these two other techniques here and provide a more comprehensive overview of the information. And as I was saying, when you actually look at this data, you can get pictures of how different parts of the brain are synchronized. So perhaps activity in 2 different parts of the brain always change at the same time. You can tell that they're really closely linked to one another. So one thing Kate has touched on is how we're using this neuroimaging data. So EEG, as Kate was saying, what you can do is we can record people's intrinsic brain activity, how, sort of what frequency their rhythmic brain activity occurs in. And then by analysing that, we can actually deliver stimulation at that same frequency. And there's been evidence that when you do deliver stimulation at the same frequency as someone's brain, you can have more pronounced effects. So that's something that could improve the outcomes of treatment. then we use this millimeter precise resolution of fMRI to actually choose the locations that we would deliver stimulation at. As Kate said, with TMS, you can just place it over a certain region. So we can move it to regions that are above the particular areas that would be suitable for that particular individual. Now, this kind of data, this kind of characterization has really important broad implications. So, as I was saying, these 3 different techniques can provide complementary information about brain activity. So the strengths of one of them complement the weaknesses of another one. Thinking quite broadly, this kind of high resolution characterization of the brain can be really helpful for understanding what is happening in Alzheimer's. So what regions of the brain are affected and how this actually leads to the memory problems that we do see. Another really important feature is actually optimizing TMS. So understanding, by looking at brain activity before and after treatment, what are the changes in brain activity that we see? Trying to understand, for people who did improve and their memory got better, what were the changes in brain activity that sort of drove those differences? And again, then trying to see, working backwards, who would actually benefit from TMS. So someone might come into a clinic in the future, record some brain activity and say, yeah, you're a perfect candidate to potentially benefit from magnetic brain stimulation. And the final aspect of the study that I wanted to cover today is something we call genotyping and blood plasma biomarkers. So essentially we take blood samples before and after the treatment begins, and we can analyze these to get some really important information. So first off, we'll be looking at a particular allele called ApoE4. So this is the strongest known genetic risk factor for Alzheimer's disease. What we can also do is look at the people's blood plasma, and there are certain proteins in there that can tell us information about what is happening in someone's brain. So without having to, take a tissue sample or do anything like that, you can just take a blood sample and get a sort of proxy marker for what's happening in the brain. So don't worry if all of these sound overly technical, but essentially the first one is called phosphorylated tau, or p-tau181. This is a protein that increases in the blood as Alzheimer's disease progresses. So if someone has a higher concentration of that, that's indicative that their Alzheimer's disease may be at a more advanced stage. The second is something called neurofilament light, and this is a protein released into the blood when brain cells or neurons are damaged. And finally, amyloid beta. So essentially, this is a proxy for determining the presence and concentration of this amyloid plaques in the brain. So the real advantage here is that this information provides another method to characterize what is happening in the brain for these people who have Alzheimer's disease. This kind of information could be used to predict who may respond to TMS, potentially from a blood sample, or for even further personalizing the treatment based on their particular characterization, whether having more amyloid in the brain may then mean that it's sort of a different parameters for treatment may be effective. But once again, this information, by combining all of this neurobiological data, we can get a much more comprehensive view about how changes in brain communication, how changes in protein markers, how these all relate to the cognitive symptoms, because in order to fully develop a truly effective treatment, you do need to understand what it is you're trying to change so that you can effectively do so. And finally, I will be passing over to our PhD student, Jackie Noonan. So just as a bit of by way of introduction here, so Jackie's PhD project will be extending our investigation to include a spectrum of cognitive functioning. So the clinical trial, which Kate mentioned, focuses on individuals with Alzheimer's disease. And as I said, the information we get from this study will be sort of one of the most comprehensive data sets collected. But moving beyond that then, this PhD project will extend to look at healthy older adults who don't have significant cognitive problems, and also people who have a condition known as mild cognitive impairment, or MCI, which in many cases can be seen as a kind of precursor to Alzheimer's disease. So what we have there is looking at the patterns of brain activity as they change from people who may have fully intact cognitive functioning to people with more of those difficulties. So overall, the project will help to improve our understanding of how changes in brain communication can actually affect cognitive functioning, and can potentially look at things like, for people who are in later life but have very, very strong memory and other things, what is the sort of pattern of brain activity that contributes to that? So with that being said, I'll now pass over to Jackie.